Hello, everyone. Welcome to the third installment of the 2021 NeLLi Symposium. To this point, we've already heard a number of incredible research talks. We've heard talks on the origin of the eukaryotic cell during the first session. Then in session number 2, we heard about current updates on the tree of life. And for today, to continue on the new lineages of life theme, we'll hear talks focused on the roles of novel lineages in biogeochemical cycles. So some quick bookkeeping, similar to the format of the previous 2 sessions, we'll listen to talks from 2 invited speakers and an additional 4 short talks. After each of the invited speakers' presentations, you can post your questions in the Q&A section. There will be no questions following each of the short talks. However, all of the speakers will be available during the breakout session following the talks. Today's moderator will be Allison Santoro. She is an associate professor in the Ecology, Evolution, and Marine Biology Department at the University of California, Santa Barbara. Dr. Santoro's work is focused on understanding the roles that microbes play in nutrient cycling within the marine environment. So without further ado, I'll hand it over to Allison so we can begin today's session. Thanks.
Thank you so much, Robert. And it's an honor to host this session, combining omics and seeing the impact of microbes on biogeochemistry, which is a topic very near and dear to my heart. Our first speaker today is Alexander Jaffe. Alex is a PhD student in the Plant and Microbial Biology Department at the University of California, Berkeley. Broadly, his work explores the evolution and function of uncultivated bacteria and archaea in the environment, with a particular emphasis on the candidate phyla radiation within the bacteria. Today, he's going to talk to us about diverse bacteria from the CPR, which shape carbon and sulfur cycling in a meromictic lake. So thanks, Alex. Looking forward to it.
Thanks so much for having me. Can you confirm that the slides look okay from your end?
Confirmed, and we can hear you great.
Okay, cool. Yeah, so hi, everyone. Thanks so much for joining us here today to hear about my work. I'm really excited to tell you about some of the stuff I've been working on for my dissertation on my favorite group of microbes, which are the candidate phyla radiation bacteria. So yeah, as Allison mentioned, I'm a 6-year graduate student in the Banfield lab at UC Berkeley. Here's a picture of our group. And if you're not familiar with our work, we're really interested in microbiomes across diverse environment types, novel lineages of life, and this is often through the lens of genome-resolved metagenomics. And some of the work that I'll describe today draws on those techniques. So a quick outline, I'll give a quick introduction to the organisms of interest for those who may not be familiar. I'll talk about some of the preliminary work on these organisms on a field site that we have, and then get into 2 case studies of interesting proteins of families of proteins in the CPR that we find in this lake, and use that as an opportunity to talk about some previous meta-analysis that I've done across ecosystems. So for a little bit of context, here we have a relatively recent construction of the tree of life. And as many of us are aware, metagenomics across many diverse environments have really greatly expanded our understanding of the diversity of lineages in the bacteria and the archaea in particular, and also some of their metabolic potential, right? What their functions can be. If you went to some of the talks last week, you might know that the position of the candidate phyla radiation bacteria in the tree of life is changing, but the point remains the same, that this is an extremely phylogenetically diverse group. So in this tree, I have them highlighted in purple with an arrow, and there are some really interesting biological features that unite them across this huge phylogenetic diversity. So in particular, that they generally speaking have these really reduced metabolic platforms, and I'll come back to that in a couple slides, and also have really small cells on the order of 0.2 microns or less. So like I alluded to in the beginning, we've studied these organisms primarily using a set of techniques called genome-resolved metagenomics. I'm sure many people here today are familiar, but just to recap and make sure we're on the same page, basically what the set of techniques involved is collecting a microbiome sample from your environment of interest. In our case, it's going to be a freshwater lake, going through extraction of genomic DNA, sequencing that genomic DNA, in our case using short read technologies, assembling those short reads into larger fragments of contiguous sequence, which we call then contigs or scaffolds, and then grouping those scaffolds together into biologically meaningful units, which we call bins, right? And these are supposed to correspond to genomic information for an organism or a population of closely related organisms that we found in a microbiome sample. And once we have those bins, we can go through and do quality filtration. We can annotate the genomes for metabolic functions of interest. We can do phylogenetics or phylogenomics, right? There are a lot of downstream applications. So using this set of techniques over the years, we've been able to attain largely complete, if not in some cases complete, genomes for these candidate phyloradiation organisms, and look into those genomes and try to understand what their metabolisms look like. So as I said, they've got a highly reduced metabolic platform and seem to get, in general, most of their energy from fermentation. They're mostly non-respiring. Using the set of techniques, it's also really interesting to see what we can't find. So by and large, these organisms are missing pathways for biosynthesis of amino acids, of lipids, of nucleotides, right? And based on these predictions and also some of the experimental work that's been done over the last 5 to 6 years, we've speculated and in some cases confirmed that these organisms are actually reliant on other members of microbial communities for some of these core building blocks of life, right? So what I'm showing you over here on the right is some really nice microscopy from 2015 showing one of these CPR organisms. In this case, it's called Archaeal-Bacteria, which you see as the tiny green cells. And in the red cells are its hosts in the oral microbiome, which is an actinobacterium. And so there's been a bunch, sort of an explosion of work like this over the last 5 or 6 years. And what the picture that's evolving is that these relationships sort of fall along a spectrum, right? In some cases, they can look more like episymbiosis. In other cases, I think we'll hear more about this next week, these relationships can look like parasitism. And the nature of the symbiosis, what's exchanged, what the dynamics are, is still very much evolving. So I think that's all to say that this is a really exciting time to be studying CPR. And a set of questions that I've been really interested in lately is to what extent their functional potential varies within environments, particularly over smaller spatial scales where there are geochemical gradients. To me, this is interesting because it gives us the potential to understand whether gene content in these organisms that are host associated is shaped more by that host relationship or it's shaped by the environment around them. The flip side of this coin, of course, is not asking how environment affects CPR, but how CPR affects their environment, right? Particularly ecosystem biochemistry, biogeochemistry. And this is where I'll spend most of my time today, given the theme of the session. So there's been a lot of work on different metabolisms in the CPR, but we still need additional systems where we can really understand how they fit into the broader community context for metabolic potential. So fortunately for us, we have a really nice model system for answering both of these questions. So we learned from some collaborators that there was a lake in central France. It's called Lac Pavin, which has a relatively high abundance of CPR organisms and also some really interesting geochemical characteristics. So this lake is what we call a meromictic lake. Basically, it means it's permanently stratified. So in the top 40 meters or so, the water is relatively oxygenated. There's a steep oxycline where this oxygen concentration falls off over a relatively short spatial scale. And then from 60 meters down to the bottom of the lake at around 90 meters, it's permanently anoxic. And due to some of the characteristics of this lake, these layers do not mix. And so they're permanently stable, more or less. These lakes in general, for a long time, have been really wonderful systems for studying microbial ecology. But I think for us, it's also a particularly exciting one because it allows us to answer some of these questions about CPR that we have, given their high abundance and also this natural geochemical gradient in the lake. So I had the privilege of going out to France during the height of fall for 3 years in a row to collect samples across this water column, this geochemical gradient, and also in the shallow sediments. So we would go out in this little raft, this boat, out to a platform and use Niskin bottles to get discrete depth samples of the water column and also do some sediment cores. And for the water column samples, we put those through a process called tangential flow filtration. This is basically that middle step here. And what that does is allow us to concentrate our sample and get at that nice microbial community that we want to use for sequencing. So once we have that, we go through the process of genome resolved metagenomics that I told you about in the beginning, and we end up with a really nice set, in this case, about 500 to 1,000 genomes from metagenomes or MAGs, right, that we can analyze and ask our biological questions of. If we look at this set of MAGs from sort of the high level, right, what does community composition look like across the lake? We see some really interesting patterns. So what I have here is basically the percentage of the microbial community at the phylum level or at the high lineage level in the water column and also the sediments, and the samples are arrayed from shallow to deep. So we start off in the shallow part of the lake and then we proceed down into the shallow sediments. I think there are a couple of interesting things to note here. One, that the community composition does change at the high level as you proceed down into the different compartments of the lake. This is not super surprising because they have such different geochemical characteristics. But I also want to point out that the candidate phyloradiation, or the lineages in it, which I have in light purple, are found in almost every sample except for some of the ones in the most shallow parts of the lake. So we are really happy to see this. And we wanted to look at this a little bit more in depth and quantify the relative abundance of each one of these CPR organisms across the compartments of the lake. So what I'm showing here is a heat map of relative abundance. I have individual CPR genomes as columns. I think there are about 100 in this case. And then again, I have samples as rows arranged from shallow to deep. And what you can see is kind of an interesting pattern that in different compartments of the lake, there appear to be distinct sets of CPR organisms that are enriched or generally associated with those compartments of the lake. So for example, in the sediment, there appear to be a block of CPR organisms down here that are more or less only found in those sediments. We also see sort of a similar pattern for the anoxic zone and to some extent for the interface. The next observation that we made was that these sets of CPR, while they might be distinct, they contain organisms from related lineages. So for example, again, in our sediment, we have organisms from the Archaeal-Bacteria, from the Bacteria, and the microgenomics, and we see some of those same organisms enriched in other compartments of the lake. So we thought this was a really good setup to test that first question that we were interested in, right? How does gene content change over compartments of the lake with different geochemical characteristics? And in some sense, we're sort of controlling for relatedness, right? I don't have too much time to dig into these data today, but the shorter answer is that we don't see much, if any, differences between the genomes, at least with the sampling that we have here. And this is consistent with some observations from other environments. An idea that I dug into in other studies is that metabolic repertoires in CPR are more shaped by broad environmental scale. So soil versus lake versus ocean versus animal microbiome. And on this scale, perhaps it's more of the specific host that determines the set of metabolic capacities for CPR. We don't have any co-cultivation work planned for this study, but we will look at some correlations and kind of assess the viability of this hypothesis. Okay, so for the rest of my talk, I did want to share specific metabolisms that may not be specifically enriched in CPR from particular compartments of the lake, but they are still very much found there. And we think that they might be involved in energy conservation for these organisms and also have some consequences potentially for ecosystem biogeochemistry. So the first case study that I want to present is one of Rubisco. You're probably familiar with this enzyme. It's considered, some people say it's one of the most abundant proteins on Earth, and it's famous for being involved in carbon fixation through the Calvin cycle. And so in looking at some of these early genomes from CPR about 8 or 9 years ago, it was really surprising that such an organism with such a reduced metabolic capacity would have such a protein that could link them into carbon cycling and carbon fixation. And so one of my first projects into the lab was to follow up on this and take a broader look at Rubisco diversity, function, and evolution in the CPR. So the first thing I wanted to do was to place the sequences we found across a broad set of CPR within sort of a broader phylogenetic context of the Rubisco superfamily. So what you're seeing here is an unrooted phylogenetic tree of Rubisco sequences across the superfamily. This is actually quite a broad superfamily with a lot of sequence and functional diversity. It contains sequences found in archaea and bacteria and eukaryotes, and actually in some cases, maybe even viruses. And these have different pathway affiliations. So just to give you a little bit of context to this tree, I'm going to point out this first clade over here on the left, which is the form 1. These are probably the ones you're most familiar with. They're found in plants, cyanobacteria, algae, some phytoplankton, and sort of involved in this classical Calvin cycle that is so well studied out there in the world. A similar thing is true of the form 2 Rubisco. That's the next clade over on the left. This is also involved in the Calvin cycle, generally speaking, and is primarily found in phototrophic bacteria like Proteobacteria. Okay, and the last one that I want to show you is a sort of diverse or sort of diffuse clade of sequences down at the bottom left. This is what we call the form 3, or in particular form 3b Rubisco. These are generally found in archaea. It's actually been shown from biochemical studies that this form of Rubisco does not function in the Calvin cycle, but instead works in what's sort of called a nucleotide assimilation pathway. So it allows these organisms to take up free nucleotides or nucleosides from the environment, incorporate a molecule of carbon dioxide with Rubisco, and turn it into a metabolic intermediate called G3P, which can sort of be easily shunted into central carbon metabolism. So this could be a really useful strategy, if you're living in sort of an early Earth where basic heterotrophs are present in microbiomes. Okay, so now to kind of get into some of the non-canonical clades that we find in CPR, I first want to point out the form 2-3, which is over here on the left. While it is closely phylogenetically related to the form 2, it does not function in the Calvin cycle, but instead appears to also be functioning in this archaeal-like nucleotide assimilation pathway. When this was discovered in the CPR, it was the first time that the distribution of this function had actually gone outside of the archaea. So this was a really exciting finding. And I should also note that there was some experimental validation of this done about 5 years ago. So one of these 2-3 sequences from the CPR was expressed and used to complement a Rubisco deletion strain of Rhodobacter rubrum, which is quite commonly used for experiments like this. And it was actually shown that it could support phototrophic growth in these organisms, which was super exciting. I want to tell you about two more, and then I'll be done. In our analyses, we also found two other clades of rubisco sequences, one on the bottom right, which is the form 3-like. These are found in CPR, and another lineage of highly metabolically reduced archaea called the DPAN, as well as a basal clade, or it appears to be basal clade of rubisco sequences called the form 3C, and these are only found in CPR so far. Of course, we wanted to get a sense for what the pathway affiliation of these new groups was. So we took a genomic context approach and looked on the original contigs where the rubisco gene was found to see what other genes were in the immediate neighborhood. And of course, particularly for the form 3C, we found that the two other genes that we need for this nucleotide assimilation pathway, DEOA and E2B2, were found in the immediate genomic context of this rubisco gene. In some cases, they were even fused together. So we think that this was pretty good validation that there was the potential for this nucleotide assimilation pathway in organisms with the form 3C and the form 3-like. Of course, it would be great to continue doing some experimental validation of these pathways in the future, as well as characterize the kinetics of some of these new rubiscos. Okay, in any case, these findings, particularly the distribution of phylogenetically distinct but functionally related rubiscos across diverse lineages of life, really prompted us to think about the overall evolution of this protein, which is so important for life on Earth. Horizontal gene transfer had been proposed to have an important role in the in the evolution of rubisco overall, and we wanted to take another look at that with our new data. So we drew on some of the new insights that we had generated to put together a new conceptual view of rubisco evolution across the tree of life. It's quite complex, and I'm not going to go through all the specifics here, but the takeaway is that you need a lot of horizontal gene transfer, including that sometimes even between domains, right? Between bacteria and archaea, between bacteria and eukaryotes, and other combinations thereof to explain this distribution of different rubisco types across far-flung lineages of life. And it's a fun place to mention that we did also find some highly divergent homologs of rubisco in some bacteriophage, which was super exciting, but they're functionally uncharacterized and they probably don't perform carbon fixation. So there's a lot more to learn here. Okay, for the second case study and the last couple of minutes of the talk, I just want to tell you about another metabolism that was discovered in some early work of CPR genomes. It was found that in some early metabolic reconstructions of CPR, I think this one is a Parku-bacteria, there were several different nickel iron hydrogenases, which you can see over on the side of the metabolic diagram, that were potentially contributing to energy conservation in CPR. And one of them, which is highlighted in yellow, was found to be most closely related to a archaeal cytoplasmic hydrogenase that was actually shown to biochemically to function in the reversible oxidation of hydrogen or the reduction of sulfur compounds like polysulfide. And this was a really exciting finding because if these enzyme complexes could also function similarly in CPR, it would be another way for these organisms to fit into sulfur cycling in ecosystems where they're found. So I took a similar approach here, and I drew on a large database of CPR genomes to situate these nickel iron hydrogenase sequences within a broader tree. So again, we have a very large superfamily of sequences with different functions. And I'm going to focus today on the group 3, in particular, sort of the region around the group 3b hydrogenases. So we made this phylogeny and actually showed that there were two clades, or appeared to be two distinct clades of nickel iron hydrogenases in the CPR, one which I called HYDE1 and one which I called HYDE2, which were distinct from, but related to anything that we had seen before. So the closest related clade that had been biochemically characterized was up here. And again, these are the group 3b hydrogenases that were found in some archaea performing that function that I mentioned earlier with hydrogen or some sulfur compounds. So we wanted to take a little bit of a closer look at the potential function in these novel clades of hydrogenase within CPR. And so we look for some of these other subunits of the group 3b hydrogenase that we would expect if it were to function in this way or to be related in this way. And we really found that only the HYDE1 sequences consistently had these three other subunits that were expected, whereas the HYDE2 sequences only had the small subunit. So this was an interesting difference, and we wanted to dig into this a little bit more. I'll explain the workflow really quickly because I think it's interesting. We took our sequences for our large subunit of the hydrogenase, or the catalytic subunit, and we sort of treated these as foci, and we went back into the contigs where these genes were encoded, and we walked out upstream and downstream of that gene for a given radius. Let's just say it was 10 open reading frames. We grab all the protein predictions in that radius, we cluster them, and then we try to assign some functional annotation, in this case using HMMs. And so what that allows us to do is to look at genomic contexts on bulk, right? Sometimes it can be difficult to do this at 20, 30, 50, 100 genomes. And I think this is a nice method to allow you to do this at scale. And so to quickly explain this plot, we have at the 0 position, the large subunit of the hydrogenase in black, And then everything to the left is going to be upstream, everything to the right is going to be downstream. And for the hide one, we sort of consistently find protein clusters associated with the 3 other subunits in the immediate upstream context. So this was nice to see, and it suggested that perhaps these genes at least had sort of the other subunits that would be required to perform this function. Of course, we would still need to do some additional biochemical characterization, but it was exciting to find this in the lake, particularly because the anoxic compartment is quite rich in sulfur compounds. We see a very different story again for hide 2, which only had that small subunit in the immediate genomic context. And so functionally speaking, we don't really know what this does. It might not even be functional at all. And I think there's an interesting evolutionary story to tell here as well. Okay, so just to relate this back to the study system, the plan is to look at some of these different metabolisms in CPR, quantify them in the different compartments of the lake, and sort of understand how they fit into this bigger picture of biogeochemical transformations that are being executed by other members, right? We have a lot of different biogeochemical transformations going on here, executed by a lot of different bacteria and archaea. And so the longer term goal is to dig into some of those nice mags that we have and think about how things interlink and what the magnitude of those processes are. So that's work that's ongoing now. Okay, and just to summarize, a couple of take-home messages. Some CPR bacteria have the potential to shape carbon and sulfur cycling, particularly in systems where they're higher abundance. The functional potential in CPR bacteria is likely not correlated with geochemistry, at least in our ecosystem, and instead the host may shape their capacities. And finally, that CPR bacteria encode novel forms of well-studied enzymes that may expand both functional diversity and our evolutionary perspectives. So with that, I just want to say a big thank you to all of you for coming out today, and to my mentors, Jill Banfield, of course, Cindy Castell, Simonetta Gribaldo, Paula Mathias Carnevali, and Marie Schulmerich, and also a big thanks to our collaborators at the University of Clermont-Auvergne for giving us access to this really cool field site and for supporting us in that, as well as a thanks to my funding sources. So with that, I think we maybe have a couple minutes for questions. Otherwise, yeah, I'll be around in the breakout room. Thanks so much.
Great, thank you, Alex. That was a great talk. Just a reminder to all our attendees to please use the Q&A function to ask your questions, and that will allow Alex to answer questions offline if we don't have time for everything. So the first question is, cool work. How did you determine which gene transfers of Rubisco were most likely in the evolutionary history of the protein complex?
Yeah, that's a really good question, because when you're working with ancient evolution, there are, of course, a lot of different possibilities, and some are more parsimonious than the others. I think in this particular case, we tried to base, in cases where it was ambiguous, where it could have been one direction or the other, we sort of tried to base it on how many sequences had been discovered in each of those groups, and basically do our best, at least conceptually, to trace back the presence of those sequences to sort of a common ancestor within a particular group. So, if we only found one of these Rubisco sequences in a clade of Archaea, let's just say, and then we found 10 in a clade of bacteria, and it appeared that there was some evolutionary link between them, it felt more parsimonious to say that that probably went from, bacteria to Archaea. But I think that's a really important question. In longer term, it would be really great to revisit this a little bit numerically. This was kind of more of a conceptual view, and obviously there are many possibilities. And so I think ultimately ascribing some probabilities to this in a more numerical framework would be a great way to look at it.
Thank you.
Yeah.
Next question is from the chat. Are there examples of viruses or microorganisms complementing host autotrophy through obligatory symbiosis?
Complementing host autotrophy. So in that case, potentially with their Rubisco gene.
Yeah, I believe that's the nature of the question.
Yeah, interesting. So I'll address the viruses first. So at least in my knowledge, we've only found this kind of odd, highly divergent homologue of Rubisco in viruses, and there haven't been any what they call bona fide, which is form 1, 2, or 3 Rubiscos that actually function in carbon fixation on viruses. And so I think if and when we find that, that would be a really, really exciting finding, because it could suggest that there might be some augmentation in that way. When it comes to CPR, I think we still don't really know what role the Rubisco gene is going to play in those host relationships. Our best guess is that it's a way for them to salvage nucleotides from their environment. So you could imagine some kind of public good that's out there in the community when a cell lyses, or maybe even when the host cell lyses, they might be able to take advantage of resources in that way. And that might be a reason why, evolutionarily speaking, they have this Rubisco. But longer term, I think that's a possibility that we'd really like to keep our minds open to. I think the nature of these symbioses, particularly for CPR and also for phage, or whatever one you want to call it, interaction, is very much an open question.
All right, our next question is, can hydrogenases use F420 instead of NADP?
Yeah, so I don't know if I have a great answer for that, as kind of my knowledge of the broader hydrogenase world is a little bit limited. I think in this particular case, we're working with such a minimal metabolic machinery that we generally sort of only see this pathway involving ferredoxin and NADP coming off of the fermentation apparatus. So I don't have a great sense for whether that's something we see in CPR. I'd imagine that should be possible somewhere else out there in the bacterial world.
Okay, we have time probably for one more question if someone wants to throw a quick question in the Q&A. Well, if not, thanks, Alex, for that great talk. It seems like, oh, wait, we have one just came in.
Okay.
Okay, this is from Anthony. For the group 3B nickel iron hydrogenases,
Sure.
Is there any correlation with the lake depth or oxygen concentration and CPR clades that encode it, or are they widespread throughout?
Yeah, that's a great question. And this is kind of what I'm interested in looking into as soon as possible. I don't have any data to support this, but I think our expectation would be that they would be more abundant in the anoxic zone of the lake where sulfur compounds, or at least at the interface where some of these sulfur compounds are pretty high, right? So this is a sulfide-rich lake. That being said, I sort of did allude to these results that suggested that there didn't appear to be, statistically speaking, major differences in the gene content between CPR, in the anoxic zone versus the interface versus the sediment. So it might not be something that has a strong statistical signal, but I'm sort of looking forward to diving into that.
Thanks for the question and thanks for the answer. If anyone has additional questions for Alex, you can type them in the Q&A and he'll be able to type answers to them. But we're going to move on to, so thank you again, Alex, great talk.
Thank you.
We're going to move on to our flash talks, and during these short talks, we're not going to have time for questions afterwards. So I'd ask you to hold your questions and then join in our breakout sessions at the end of our time together today. So our first flash talk today is Meng Li from Shenzhen University. He's a professor there, and his research is centered around aquatic microbial ecology. So take it away, Meng.
Thank you. Can you hear me?
Yep, sounds great, and we can see your slides great. Thanks.
Okay, so today my talk is about the Asgard-Archaea. So because for the slide I'm showing, actually it should be the second slide. Actually, I'm not quite sure how it's moved directly. Okay, let me see. Okay, today the title is my talk is like Asgard-Archaea active in the marine sediment carbon cycling. So as we know, Archaea is the third domain of life. So since like 1977, and recently, because the hypersequencing technology developing very quickly, so the Archaea diversity expanded very quickly. So you can see in the left panel, before the 2002, there are only two phylum of Archaea was identified. It's called the Archaeota and the Archaeota. And after 2002 to 2011, there have one superphylum called the TACK was identified. Now already like more than 36 different phylum have identified. They are including 3 superphylum. We call it is TACK, Asgard, and the Dipan. And also another one is like Archaeota. Among them, the Asgard-Archaea is one of the superstar. Why we call the Asgard-Archaea is a superstar? Because the Asgard-Archaea is considered the close knowing prokaryote relative of eukaryote. And then from this phylogenetic tree, we can see that the Asgard-Archaea is most closely related to the eukaryote. It means that change our traditional view about the 3 domain. Now we can come back to the 2 domain. So in 2012, the Japanese scientists have first isolated the first Asgard-Archaea called Archaeal-Bacterial. So and then most of people are focused on those like evolutionary insight of those Archaea, their relationship between the Asgard and the eukaryote and the Archaea. However, from my group, we majorly focus on the metabolism. Actually, Asgard-Archaea was first identified or first reported in early of 1999. At that time, they are not calling the Archaea, they are actually calling the MBGB or the DSAG. They are, I think currently those two groups are belong to the Lockheed-Archaea. From the left panel, we can see that they are really abundant in the natural environment, especially in some marine sediment. So at that moment, people already consider this archaea may play important role in the biogeochemical cycling. And lately, using the metagenomic and the information, we got a lot of those Asgard genome, and then based on the genome, we can predict their metabolism. So we can see that scientists already proposed that the Asgard-Archaea may mesotrophic or acidotrophic or phototrophic. Some group maybe can anaerobic hydrocarbon oxidation. So we are wondering, ask the question, is that in the in situ activity of those Asgard-Archaea and their physiology, whether especially whether they are doing a really good the big loss in the carbon cycling. So based on those scientific questions, we are going to do our, I would like to introduce a field of work of my research group. First, we want to identify the diversity of those Asgard-Archaea. We firstly collect those 16 sRNA gene sequence from the database. So we totally got 10,000 of the sequence, and then those like sequence is up to 800 base pair. Then we can cluster them, and then we can got more than 451 OTU. And then from those OTU, we can identify there are more than 10 phylum of those Asgard-Archaea. And especially all those Archaea, all those 16 sRNA genes are expressed in the environment, and most of them are mainly identified in the saline sediment. And especially we need to point out that different phylum of those Archaea are following a biogeographic distribution pattern. We also using the Earth project data to add distribution of Archaea from Earth micro project, and found that Archaea they widely distributed, especially the. And then we also analyze their relative abundance. We can find that the Lockheed-Archaea and the Solar-Archaea are the most abundant Asgard-Archaea. Then, based on those information, we try to collect sediment sample from different environments, including the coastal sediment, hot spring, and also the hydrothermal vent. We use the metagenomic approach. We totally got 184 genome of those Asgard-Archaea. Among them, there are 75 are new MAG. Based on those like 70 new Asgard-Archaea MAG, we totally identified 7 new phylum, including one is called Gerd Archaea from the major from those like mangrove sediment. Another 6 different Asgard-Archaea was identified in different environments. They're including the Helmholtz Archaea, Border Archaea, Bola, and Kela, and the Holda Archaea. Especially, we also named one called is Wukong Archaea. On the right panel, we can see that there are phylogenetic analysis of those different phylum. And especially I need to point out is that Wukong Archaea is not closely related to others. They are not belong to the Helmholtz Archaea. So based on those genome, we also analyze their metabolism, because my group is major focus on the metabolism. We try to analyze their carbohydrate metabolism, the methane and the carbohydrate metabolism, and also their whether they are anaerobic or anaerobic. There are also the nitrogen metabolism and the sulfur metabolism. Those are indicating the gene present or not present in the MAG. So as these find classify Archaea into 3 different metabolism group. One is called the heterotrophic Sol acuta, Homeda acuta, Odin and the Broda lock the Hamdale. One is called the active aerobic heterotrophy. Those three groups, they are part resistant to oxygen, contain the aerobic pathway. The group is we find type 4 hydrogen in the genome, and we consider they are accumulated. So based on methylation, so the in situ activity collect the sample and metatrans analyze, and we found those pathways involve carbon fixation cell and the protein degradation are transcriptional in the in addition identify high abundance like a transcript and the carbohydrate. We also do an experiment cooperator. We use the top. We try carbon 13 and as the substrate incubation in the lab. So we provide the and the electron accept long time incubation in the carbon 13 in the set in addition measuring those DNA in the sample. We two group of Lockheed sample. One is B and the Lockheed 2B. A certain they are also response to the organic composition. So in Lockheed acuta are metabolism different. To identify, we also doing the metatranscriptome by those two groups. The carbon metabolism reconstruction, we can find that appear to the mesotrophic participated in degradation the lignin and the humic acid, similarly carbon dioxide, the heterotrophic used. Why the prefer using the inorganic mark and degrading the mass the incubation. Based on those methods, form a two model subgroup of. For this one, they are possibly using those like lignin and the why for the Lockheed prefer using the carbon, the inorganic carbon. So based on those information, actually can say that are accepted in the carbon because the time limit field taken mass 7 new Asgard different carbon metabolism transcriptomy that all knowing can able of degrading including peptides amino acid.
Hi, we're having a lot of trouble with your sound. And I think we're also probably out of time for this presentation. This looks like really interesting findings, but I think maybe people could follow up with you in the breakout session. Hopefully you'll be able to stay on afterwards after that.
Yeah, thank you.
Yeah, thank you very much.
Yeah. Okay.
All right.
That's my acknowledgement.
Okay, yeah.
Thank you.
Thank you.
Yeah.
Okay, our next speaker today is joining us where it's almost 11 o'clock at night, I believe. Maliheh Mehrshad. She's at the Department of Aquatic Sciences and Assessment at Swedish University of Agricultural Sciences. She uses multi-omics analyses to study metabolisms and interactions of aquatic microbial communities in groundwater and freshwater. So thanks for joining us, Maliheh.
Hi, do you have my slides?
Yep, everything looks great and we can hear you. Take it away.
Okay. Hi, everyone. I'm going to talk about how energy efficiency and biological interaction can define the core microbiome of deep oligotrophic groundwater. Looking at the distribution of biomass on the planet Earth, you can see that a lot of biomass is deposited actually in deep subsurface. And more interestingly for me as a microbiologist is that most of this biomass in deep groundwater is microbial. So microbes are actually the biome in charge in deep subsurface. And among these different subsurface environments, I'm studying groundwater, and this big biomass is deposited into small cells. So we have an approximately 5 to 10 to 27 bacteria and archaea that are thriving in groundwater. Studying groundwater is always difficult because you need access points, and it's not easy to find good access points to deep groundwater. Here I plotted on the map all the points that we have access to oligotrophic deep groundwater around the Earth. In red, you can see the depth of those samples, and underneath I put the amount of data we have available for those data sets. And at first look, you can see how patchy our view of deep groundwater is. And then if you add the factor of depth, we know that is even more patchy. So we don't have the possibility to answer a lot of ecoevolutionary questions about these habitats. But luckily here in Sweden, we got access to two points that we can sample from deep groundwater. One through Sweden in Aspo Hardlock Laboratory, and one in Finland in Olkiluoto Islands. So through the collaboration of these three labs, they collected many samples from these locations. And this is like a really tedious task because it takes a lot of time to collect samples from these locations. They have generated 44 metagenomes, and we have generated a lot of single cell amplified genomes and 9 metatranscriptomes from different boreholes from 170 to 528 meters below surface. We analyzed all these metagenomes and single cell amplified genomes and generated genomes from them. We have almost 1,300 genomes, good quality genomes from them, and we call it Phenoscandian Sheet Genomic Database. When we cluster them at the operational definition of a species for bacteria and archaea, like 95% average nucleotide identity and 70% coverage, we get 598 genome clusters, which is a good diversity for deep groundwater that is like highly oligotrophic. But how diverse they are when I put them in the tree of archaea and bacteria together with all other known genomes, and the FSGD genomes are shown in red, you can see that they cover a big diversity of the bacteria and archaea. So our first question was whether we will see any genome that is present in both these locations. Because this study of deep groundwater should be always done hand in hand with geology, and most times the samples we get, they are coming from so many different geologies and we cannot compare these. But we are lucky because those access points we have, they are in the same bedrock with similar geology. So in this heat map, you see each column is a genome cluster and each row is a different metagenome from different location and different borehole. In green, you see presence, and in yellow, the absence. Studying this heat map, we realized that we have 165 mags and sags that are common between both locations, and they are clustering into 73 genome clusters. They are highly diverse because we have 2 archaeal phyla and 13 bacterial phyla in these common groups. And this is interesting because now we can speculate on how they have converged in these 2 different locations. Their high phylogenetic diversity suggests an ecological convergence, probably due to availability of nutrients and reducing agents, but we cannot still refute the possibility of an evolutionary convergence. And we have studied that in more detail, but I will not discuss it here. And in the below panel, in this heat map, you see in brown those genome clusters that we actually see them being transcribed in our transcriptomes. This was interesting to see quite a lot of these genomes being actually transcribed. So we decided to study them in more detail. We mapped all those transcriptomes against these genomes and separated those that are highly transcribing. We had to put an arbitrary threshold, like the nightmare of every bioinformatician, but we decided to go for 10,000 or more transcripts per million. And by doing that, we found 86 genome clusters that were highly transcribing in 17 different phyla. We were not expecting to see Pathocybacteria among these highly transcribing groups, but you now know them really well from the first presentation. They have really reduced genomes and are suggested to be symbiotic, and it is interesting to see them highly transcribing in a deep oligotrophic groundwater. We found 4 of these genome clusters to be able to produce acetate from acetyl coenzyme A. And then I will come back to this, that why I think this is interesting. 2 other groups that I'm going to discuss today is the Sulfobacteriota and then phylum UBA9089. It still didn't get its own name. It has a code. But they are interesting because phylum UBA9089 is a group that is specific for groundwater. So all reported genomes are originating from groundwater. We had 5 species in our FS3D genomes. Their genome size ranges from 2.3 to 3.5 megabase per and the GC content of 34 to 44%. The other phyla is the Sulfobacteriota, has a much higher diversity, 165 species, and 15 of them among the highly transcribing genomes. Their genome size ranges from 1.8 to 7.4, and the GC content of 35 to 67%. Their interesting metabolism was that they both have the stimulatory sulfate reduction and they have Wood-Lyondell pathway. Sulfate reducers run Wood-Lyondell pathway, that is like a carbon fixation in reverse, and they oxidize acetate by running this in reverse. And that is interesting because that gives the cell the possibility to use acetate as a carbon and electron source. And makes it even more interesting because this process, when it's run on the reverse, is thermodynamically unfavorable. But coupling it with a favorable process like sulfate reduction allows these cells to use acetate. And it is connecting back to Phytacibacteria being highly abundant because highly expressing because they are producing acetate, and this can provide the possibility of a reciprocal partnership between Phytacibacteria and these sulfate reducers that are using that compound. We have done several other analyses that I cannot discuss in this short time, but we find out that cooperation and energy efficiency are helping deep groundwater microbes to diversify, and we show proof for that instead of a lifestyle where microbes are predominantly investing in functions related to maintenance and survival, in deep groundwater, a cooperative and episodic lifestyle enabled by reversible bacteriostatic functions ensure the subsistence of microbes. And by that, I want to thank you for your attention and thank my collaborators.
Thank you so much, Maliheh. What a neat data set. We're going to move right on. Our next speaker is Joe Vineis. He's at Northeastern University, where he's a student in Jen Bowen's lab. He uses omics techniques to identify microbial contributions to biogeochemistry in salt marsh sediments. Joe, how is your internet working today?
You tell me.
We're looking good. Take it away.
Good?
Yep.
Okay. All right. Yeah. So thanks, Allison, and thanks to the organizers. I'm just so happy to be part of this symposium. And today, I'd just like to tell you a bit about one small story in the broader stories that I've been developing in salt marsh sediments. And this one is regarding TAO6 and that it inhabits really diverse nutrient space and functionally diverse microbial neighborhood. Oh. Let's see. So for those of you who are not familiar with salt marshes, they're really highly productive grasslands. They store carbon at high rates, comparable or more than most other terrestrial systems on Earth. And they also support large and diverse populations of microbial organisms that use really diverse metabolisms, especially anaerobic metabolisms, due to the nature of the sediment. And so what I want to just share with you really quickly is the structure of the sediment. So on the left-hand side of the slide, you see sort of a theoretical diagram of the carbon and how it's structured within the sediment. So this diagram really goes from 0 to 250 centimeters. The shallow sediment at the top of the diagram shows that there are tidal and plant sources of carbon. So in this rhizosphere here in the shallow sediment, there are tons of root exudates, and oxygen is released by plant roots as well to perform, in essence, allows the microbial community to conduct large amounts of metabolically diverse activities such as sulfate reduction, sulfur oxidation, denitrification. And just below the rhizosphere, so around 40 centimeters where plant roots sort of stop growing, we have sort of an intermediate complexity of carbon and nutrient availability. So there we see a lot of fermentation. There's chemoautotrophy within the sediment. And beyond that, where there really are no tidal inputs and no plant inputs remaining, within this deeper sediment, it's primarily oligotrophic. So there's very low complexity of the organic matter. We largely have fermentation and chemoautotrophy. And so the questions that I've been thinking about and working within the system are, how are these microbes structured along the gradient? What nutrients and carbon sources do they influence? And what are the interactions among microbes that influence the biogeochemical reactions? Primarily carbon. We think about nitrogen and sulfur as well within these systems. But really, this is the niche space, right? So what are the niche space of each individual organism, and how are they distributed and interacting with one another to affect broader cycles of carbon and nitrogen and sulfur? Okay, so to address these types of questions, we need to, of course, take large sediment samples. So we pulled 6 push cores from the sediment and partitioned them into different depth fractions. And we took 6 total samples, as I said, or 6 total cores, as I said, and partitioned them by depth into 55 total samples. So each one of these bars here represents a depth fraction that we sampled from these larger cores. And then thanks to community sequencing from JGI, the proposal IDs right here, we were able to use large effort of genome resolved metagenomics to recover over 2,000 mags from the sediment, which are on largely medium to high quality mags. And so this is a ribosomal protein tree of all of the mags that we were able to recover. These are redundant mags, they haven't been dereplicated. But there are some really interesting groups. I think to many of us here, we've seen several slides with these wonderful groups here: Chloroflexota, Planktomycetota, these Peitzi bacteria, all so interesting. And really, when I thought about talking at the symposium, it's really just a feast of you could pick any one of these groups to come up with interesting stories to talk about. But I chose TA06 because of some really interesting aspect to that particular group of mags that we isolated. So because I isolated mags on a per sample basis and not through co-assembly, I was able to recover 24 independent mags from the sediment. And so this is, again, just a theoretical image of an image of the sediment core, and each one of these arrows indicates where we recovered a TA06 mag. And so all of these 24 mags that we recovered are summarized here in this table, just so you can see that largely all of them are 92% complete and 7% redundant. And when we dereplicate these at 70% length and 90% identity, we end up with just a single cluster of organisms. And so that, to me, was really surprising and super interesting, because how is that one really closely related group of organisms or population inhabiting this entire niche space, which has very diverse sources of carbon and electron acceptors and so on. So although we were able to isolate them, it's also important to look at their relative abundance. And what we see here is that they were very patchily distributed, or only abundant in 1 or 2 samples in the shallower sediments, and more broadly, we find them in this deeper oligotrophic environment. And so this indicates, the likelihood of its niche space is probably this oligotrophic, deep carbon or deeper region of the sediment core. And so the second question that I really was curious about with these TA06 is, what are the assemblage of microbial taxa that's around TA06? So who does it associate with? What are the metabolisms of those organisms that it associates with? And so I used, although messy and not always accurate, I used correlation coefficients and was able to determine that there are 49 taxa that were significantly correlated with this singular dereplicated TA06. And in this example, you're seeing the Pate-side bacteria in TiO6. So this is one individual mag and how it correlates with this particular really interesting organism. And so that's just to give some more support that although these correlations are somewhat spurious at times, there's still probably some likelihood that there are interesting relationships between these organisms. So the next thing that I was really curious about was if they co-occur together, what's the functional space like? So are all of the organisms that are co-occurring with TiO6, are they all doing similar functions, or are they in a functionally diverse space? So I built a functional network here, and each one of these small gray dots indicates a function. And the larger dots indicate a mag, and connections between the larger dot and the smaller function indicates that that mag contains a function. The letter within each of these larger mag representation dots indicates whether it's recovered from mid, deep, or shallow sediment. And so here's our TiO6 mag, and the take-home message of this broad functional network is that TiO6 is really found or seems to co-occur with organisms that are functionally diverse. And in general, these Planktomycetota seem to really enjoy the company of TiO6, and they're somewhat complementary in function. So Planktomycetota are largely deriving carbon sources from cellulose, hemicellulose, and they have a really interesting wood-lungol pathway, which is a story for another time. They also co-occur with the Chloroflexota, which are conducting wood-lungol aerophoboda. They're also wood-lungol potentially metabolizing organisms, Desulfobacterota as well. So this really just opens the door for some really more interesting questions to ask regarding TiO6 and how it survives within the sediment and operates at a broader level in carbon cycling with all of these other really interesting organisms. So this is a very TiO6 sort of centric story, and you could do this with any other organism. And I think it's a really interesting way to sort of approach and think about the neighborhood of microbial taxa that you're interested in and how they potentially work in synergy to cycle important nutrients in your environment. So with that, I'll just thank Jen Bowen, my advisor, and collaborator, Ashley Bilseco, and thank all of you.
Great, thank you, Joe. Lots of room for questions there later on. We'll move right on to our next talk. Our next flash talk is by Eddie Whip. She's a postdoctoral scholar working with Kate Zelnina in the Environmental Genomics and Systems Biology Division at Lawrence Berkeley National Laboratory. Eddie, are you ready with your slides? Great.
Thank you, yes. So today I'll be talking about the roles of plant-archaea interactions and how they relate to driving soil nitrogen availability. So I'll briefly just touch upon giving you more of an introduction of archaea and the ways they're involved in nutrient cycling, and then go into what roles plant-archaea interactions play in nitrogen provisioning. So the ways in which we're looking to answer this question. And then sharing with you some preliminary discoveries about the character of how what it means when archaea associate with plants in terms of plant growth and nitrogen and carbon relations, and provide you with a few key takeaways. And so you can call archaea this domain of the mysterious, just in how they're pretty unique, but not very well understood. A lot of research has focused on these extremophiles from hot springs and deep sea endothermal vents, and provided us with some really exciting advances in how we understand enzyme biology and kind of living in extreme environments. And more recently, they've kind of moved archaea from obscurity to greater knowledge of their ubiquity. And so first being characterized in 1977, we have up to present day a much better awareness of how archaea inhabit many different spheres of life and locations. And they comprise, in particular, up to 17% of soils, which I'm particularly interested is how they relate to plant growth and just the overall community functioning there. And we also have found that they are present in plant rhizospheres. And so some of the unique and influential traits of Archaea that we are uncovering, as you're learning from different talks as well, we know that they're involved in carbon assimilation and carbon fixation, organic carbon mineralization, methanogenesis, as well as nitrification, nitrogen fixation in a few instances, and their metabolite, secondary metabolite production is particularly interesting, and there's a lot more to characterize of that, but we're noticing that they can produce ones that interact with plants and influence their growth. And so for today, just to kind of, I'll briefly with you to discuss nitrification. And so this equation relates to the conversion of ammonium to nitrate. And this is a particularly important process in nitrogen cycling, or has received a lot of attention. And so for reasons why it's important, we'll kind of connect it back to the general nitrogen cycle, overall nitrogen cycling. And so they fit into one of the three main stages here. And it's of relevance when we think about climate change today, as well as the degree of pollution that's a lot of agriculture is contributing to, where it's been such a major advancement in society to have nitrogen, synthetic nitrogen fertilizers to increase crop growth. This has come at a great cost, in part due to nitrification, where we lose a lot of the nitrogen that we apply to the soils based on the conversion into nitrate. This is a negatively charged particle, and soil is generally predominant is overall negatively charged. We lose a lot of this with seeping into our waterways. And so there's also implications of eutrophication and ocean acidification, as well as the production of nitrous oxide is one of a major climate change contributor, where it's about 300 times worse than CO2 accumulation in terms of damage done. And we know that with inhibiting nitrification, not only can we reduce soil nitrogen losses and greenhouse gas emissions, we can increase this nitrogen fertilization use efficiency. And so, and particularly as well, when thinking about plants and connecting this to carbon cycling, we know that through a process called rhizosphere priming, where it's very important to consider how nitrifiers and these AOA or Archaea that do nitrification, is that when you reduce carbon sequestration, or there's reduced carbon sequestration when roots mine for organic nitrogen. So when there isn't high amounts of nitrogen otherwise available, soil organic matter can be broken down, and this can free up nitrogen as well as release CO2 into the atmosphere. And so I'm particularly interested, and we'll explore today with you, how do interactions between key nitrogen cycling Archaea and plants, what is the nature of their character, of their activity, and how do they influence nitrification? And so particularly ammonia oxidizing Archaea are one of the key drivers of nitrification. So we know that there's a kind of a nice diversity of these ammonia oxidizing Archaea within this group, the Thermoarchaeota. And I'm particularly using a strain that is Nitrosophora vienensis. And we know that these AOA, or ammonia oxidizing Archaea, are present in soils at 1 to 5%. We know they're found in rhizospheres, and they contribute to this rate limiting step in nitrification, so the conversion of ammonium into nitrite. And so I am curious about what roles do Plant-Archaea interactions play in the soil nitrogen provisioning. And so with my work, I'm determining the mechanisms underlying changes in AOA nitrification, as well as other plant growth phenotypes and AOA activity. So I'm thinking that AOA activity modulates plant growth and root exudate composition, and that plants will regulate AOA activity by production of nitrification inhibitors in their exudates, which can then be of importance in agricultural systems to reduce nitrogen loss. And so I'm using these fabricated ecosystems, or ecofabs they're termed, to test how AOA and the anensis impacts the growth and nitrogen uptake of biofuels crop, switchgrass. And so looking here just generally to determine how does interaction with the plant influence nitrification, so measuring nitrite levels, as well as thinking about ammonia and nitrate, as well as then looking at the metabolites that are produced and the transcripts from AOA that will give us more insights into the nature of their interaction. So here I'm going to show you that switchgrass biomass increases with AOA inoculation. We generally see that both biomass and plant height are influenced. And so here I have under nitrogen replete and nitrogen limited conditions showing you plant total fresh weight here. And we see that with Archaea, so switchgrass with Archaea is the dark green, and this light green is switchgrass alone. And then we also on the x-axis, to back up a little bit, is the days post-inoculation with AOA. So we have 7 days after inoculation and then 15 days after inoculation with AOA, just measuring how plant growth is impacted. And by the second time point, so having about 2 weeks with the Archaea, we see that there's this increase in switchgrass biomass. Under nitrogen limited conditions, we also see this increase. And this is somewhat surprising and suggests that AOA might have some kind of mechanism of promoting plant growth. And so perhaps if there's increased photosynthetic activity from crosstalk between the plant and AOA. And so in the exudates, we actually characterize the total organic carbon, and we find that there's greater amounts of organic carbon when switchgrass is in the presence of AOA. So here on the y-axis, we have total organic carbon and parts per million. And then on the x-axis, we have AOA alone, and then AOA with switchgrass and switchgrass alone. And we see that there is this uptake, increase in organic carbon. So this potentially might be due in part by the ability of AOA to fix carbon. So just generally more carbon being fixed by AOA in these treatments, that as well as it can indicate, suggest that there's this plant microbial crosstalk. And I'm really curious about these mechanisms involved with this, and some of the, in this recent paper, are some of the potential mechanisms to explain this. And through transcriptomic and metabolomic analysis, we can kind of get at some of what might be going on here, testing these different classes of interactions. And so with AOA growth being impacted, I'll just lastly show you that we find that there's some interesting patterns of AOA activity and nitrification. So here I'm showing you under nitrogen replete conditions, just with the Archaea alone, we have an increase and a stabilization of their nitrification. But with switchgrass, there seems to be a stimulatory effect and then later an inhibition period. And this is also repeated in nitrogen limited conditions. And so as I showed you, just said that there's this increase stimulatory effect, and we're really curious about activity and metabolites that correspond to these. I'm going to just jump to the summary then of what I've shown you so far of talking about soil microbial communities and Archaea, that we don't know much about them. And by thinking about plant traits and nitrification, there are a lot of interesting avenues to explore. And it's interesting here that we find with our specific strain, there's this increase in plant growth. There's a lot of, so thinking about metabolites and produced by switchgrass, as well as just what are the metabolites being produced by Archaea involved in this interaction. And also thinking about different AOA cultures and that we've enriched from different soils to also test how they interact with Archaea. So just I wanted to acknowledge everybody who's been a part of this work, my advisor, Kate Salninia, and Krista Slepper was involved in actually giving us the strain that I've shown you the results from. And yeah, the Northern Lab at the Lawrence Berkeley National Labs has provided a lot of great resources and commentary. And so I just want to thank you all, and I'm available for questions. There was a lot of things I left out here with troubleshooting and other methods, and happy to address these in the Q&A and beyond. Thank you.
Thank you so much, Eddie. If you have questions for Eddie, you can join us in the breakout sessions after our last talk, which is right now. So I'll introduce our final speaker, who probably needs no introduction to this audience. Victoria Orphan is the James Irvine Professor of Environmental Science and Geobiology at CalTech, where she holds the Davis Leadership Chair and is also the director of the Center for Environmental Microbial Interactions. She's the recipient of a MacArthur Fellowship. Her research focuses on microbial communities and marine sediments, especially those associated with methane cycling. And if you have questions for Victoria, we'll answer those in plenary here, so you can put those questions in the Q&A, and I'll moderate that afterwards. So if you're ready, Victoria, you can go ahead and start sharing your slides.
Great. All right, thank you, Allison. Let's see if I can get this.
I should also mention that Victoria is an alumna of the university where I am. She's a Gaucho. So.
Go Gauchos. Fantastic. All right, Allison, can you see my slides?
Yeah, I can hear and see you great.
Fantastic. All right, thanks for that nice introduction. I'm happy to talk to you about one of the things that I love most about my job, and that is working with syntrophic associations in the deep sea. So I'm going to tell you about what we've been learning about the ecophysiology and the specific types of symbiotic interactions that occur between archaea and bacteria, and more recently, the role of viruses in these special places in the deep sea called methane seeps. And these are places where methane is ejecting to the seafloor, and they are ideal natural laboratories to study this process of anaerobic oxidation of methane, where you have archaea that oxidize methane in symbiosis with sulfate-reducing bacteria that use sulfate from seawater. And so what you can see on the title slide here is a fluorescence in situ hybridization image of these multicellular consortia as they exist in the seep sediment. And so we've been interested in understanding a lot about the ecology, evolution, and biogeochemical roles of these organisms in the environment. And we care about this from a broader standpoint because they are basically the main way that methane is oxidized in anaerobic sediments in the ocean. And they are largely responsible for keeping that methane out of the atmosphere. And so they play a huge role. And like many microorganisms, they have yet to be cultivated, at least in pure culture. And so a lot of what we've learned about them is using these omic type of methods, as well as other types of geochemical and microscopy techniques to understand them in the context of the sediment environment. And so again, if we look at a little bit of this sediment that was collected from a deep sea seep, and we zoom in on a few of these different consortia, we see that they represent a huge diversity of different morphotypes. Sometimes they are in affiliation with different organisms. Sometimes they have very well mixed associations. Other times they're more clumped in their distribution of those cells. And we've been interested in what drives the specificity of these partnerships, what constrains the spatial relationships between these organisms, and really how do we find so much diversity of these different organisms, presumably all doing this process of anaerobic oxidation of methane, all co-occurring within the same kinds of samples. So what we do know about these organisms largely is that it appears that the symbiosis is driven through extracellular electron transfer, where methane-derived electrons are directly passaged outside of the archaeal cells and taken up by the sulfate-reducing bacterial partner. And so there's a number of different publications that are listed here. I'm not going to have time to really go into the details of this, but we think this is one of the main ways now that this symbiosis works in the marine environment. We also have a lot of information we've been gathering from the different groups of these methane oxidizing archaea, all called ANMI, of different flavors. And they're highly diverse, in some cases representing different order level distinctions. And collecting this information at the genome level is allowing us to do comparative work to look for the similarities and differences, both among the different groups of these methane oxidizing archaea, as well as comparing them to their closely related methanogenic relatives. And correspondingly, we also see a similar level of diversity with the known syntrophic sulfate-reducing bacterial partners, which are largely part of the Desulfobacterota groups here. And so they're all the ones that are these collapsed clades that are colored all with the Seep designation in front of them. And so some of the questions that we have are, what is the specificity of pairings within individual consortia? And so to do this, we were applying sort of classical statistical correlation analysis just from looking at 16S ribosomal RNA genes that were recovered from the environment. And this is a recent study that was done looking at over 300 methane seep sediment samples from the Costa Rica area, and just asking what are the strongest correlations that we see. And from that, there was some nice groupings of different types of methane oxidizing archaea, the ANMI2C grouping with the Seep SRB1A cluster. And also what was interesting to us were these members of the ANMI2B up here that was associated with a group that we previously hadn't affiliated with a symbiotic sulfate-reducing bacterial process. So this is the Seep SRB1G clade up here. And so from this, we can sort of take this prediction of co-associations that just come from 16S genes recovered from the environment and independently test this using other types of techniques. And one of the ways that we can do this is by individually sorting individual consortia and looking at the co-association. And so, so this is work that was largely led by a former postdoc, Roland Hatzenpickler, in the lab. And to do this, we were using a technique known as bioorthogonal non-canonical amino acid tagging, or BONCAT. And this allows us to fluorescently label cells that are active in protein synthesis, such as this aggregate that's shown here in green. And so based on that fluorescent signal, we're able to fluorescently sort active consortia from incubations where we've stimulated them with methane. And so Roland was able to do this in collaboration with the folks at the JGI and the Single Cell Institute, led by Tanya Wojcicki. And from this, we were able to sort individual aggregates, MDA amplify them, and then ask the simple question of correspondence of 16S ribosomal RNAs within those aggregates, as well as get information about the genomes from both partners. And so in Roland's study, he was just looking at the 16S ribosomal RNA genes initially. And so what he was able to show was that the Seep SRB1A, as predicted, was associated both with the ANMI2C as well as the 2A, and that the ANMI2B, shown here in green, did form associations with this new group of Seep SRB1G. And the thing that's interesting here is that these samples came from a totally different Seep environment. So the work from the before was done off of the coast of Costa Rica, and now we're looking at the coast of methane seeps off the coast of Oregon. Yet we're still seeing these same sort of patterns emerge in terms of the specificity of the partnerships. Okay. So you can also use these 16S ribosomal RNA genes, of course, to design fluorescence in situ hybridization assays and directly visually confirm the association. And this is what we did designing a new probe for the Seep SRB1G and showed that it does indeed form these tight physical associations with the ANMI2B. And so this provided yet another independent line of evidence of this specific new type of association that is common in the Seep environment. Now, from this data, we were able to also start to look at the genomes of this Seep SRB1G, and what we found was that it encoded some interesting features that we wanted to explore further. And so I'll get into that in a second, but right now I wanted to look at the genomic variation we can see between the ANMI2A and the ANMI2B groups here. And so each line now represents sort of a linearized fraction of the genome where we have basically colored lines of the genes, orthologs that are in common between these different groups. And so we can see between the ANMI2A and the 2B, there's a group that is core across all of them, but we also see there's quite a bit of variation. In particular, clusters of genes within the 2A that are not found within the 2B group here, as well as other types of interesting genes like this PPK polyphosphate kinase that was found exclusively within or largely within the ANMI2B, but not associated with the ANMI2A. So if we want to look at the diversity within just the 2B single aggregated sorted genomes, what we find was that there was actually even some subclustering within these groups here. So each of these organisms are sort of related at the level of 98% ANI. And you can just see here that there's distinct clusters of genes that are unique to different groupings or subclusters of these ANMI2Bs that are all coexisting and all recovered from the same sample. So we're still looking at what's encoded in these different clusters here, but this may give us some insights into specificity and niche differentiation among these different groups. So let's look at this PPK, so the polyphosphate kinase, which is predicted to be involved in polyphosphate synthesis. And so if we look at the genes in the neighborhood around that PPK ACE, we find high conservation of things that are related to phosphate transport, as well as phosphate regulation that's conserved amongst all of the ANMI2B genomes that we sorted. So this gives us some insight into what's going on, but we want to go beyond just looking at genomic predictions to what the organisms are doing themselves. And so to do this, we tried to combine fluorescence in situ hybridization with other types of imaging techniques. And of course, things like polyphosphate in some cases are hard to detect in environmental organisms and frequently require ultrastructural information that are provided from electron microscopy techniques. And so here we were able to combine fluorescence in situ hybridization using a FISH probe targeting the ANMI2B and combine it with EM-based analyses. And what we find are these large intracellular granules that were reminiscent of what's been described in terms of polyphosphate granules. And so we can look at these from a chemical perspective to get a sense of what these granules are made of. And we can see here in the secondary ion mass spectrometry image that they are not made of nitrogen or sulfur, but using other types of techniques like eels, we can resolve that indeed these intracellular granules are enriched in phosphorus as well as iron, so consistent with a polyphosphate granule. And these granules seem to be unique to the ANMI2B Archaea and were not observed in the co-associated sulfate-reducing bacteria. So looking at these polyphosphate granules, again, we looked at a lot of different types of aggregates, and they seem to be unique to organisms that we had identified as being the ANMI2B. And we can further use 3D reconstructions using a technique known as serial block face imaging to section each individual aggregate and then reconstruct the biovolumes of the different partners of ANMI2B and their Seep SRB 1G partners. And so in doing this, this gives us information about just how much of the biovolume of the cell is comprised of these large polyphosphate granules, which are about 200 nanometers in diameter. And each cell largely had either one or two of these large polyphosphate granules. And the interesting thing that we saw was that there would seem to be some relationship between the size of the granule and the numbers of these granules within the cell. So the amount of biovolume represented either by one big one or a bunch of small ones was relatively consistent across cells. If we compare across those biovolumes, we see that these polyphosphate granules make up about 1.5% of the total cell volumes. And we can compare this with some research done on other types of related methanogens where polyphosphate granules are also produced. And in this case, they represent about 2.2% of the cell volume. So this is a pretty significant phosphate store within these ANMI2B. Now, we don't, of course, know the function of these polyphosphate granules yet, but if we think about it from the context of just energy, pure energy stored, it's something on the order of about 200 millimolars of ATP equivalents per cell, which would be a real benefit for an organism that's basically eking out a living doing sulfate-coupled methane oxidation. Okay, so let's look at the also the syntrophic Seep SRB 1G partners within this association. And so this is work that's being done by a postdoc in my group, Ranjani. And what's shown here basically is a genome tree of all of the major groups of syntrophic bacterial partners that we know of these Archaea. And we're going to focus in here on the Seep SRB 1G, which, like the ANMI2B, is less diverse than some of the other organisms, sort of on the level of 97% ANI score here. And this transcends across different environments. So each one of the symbols here represents a genome that was assembled from a different Seep around the world. So a lot less diversity within the ANMI or the Seep SRB 1G. And so looking at the genomes of these organisms, I mentioned earlier that we saw evidence of a nitrogenase. And this was interesting to us because we have been looking at the role of nitrogen fixation in these methane seep ecosystems. And by looking at the nitrogenase genes, we were able to go back to some of our earlier data of looking at expression of nitrogenase within the seep environment. And what we found was that one of the highest expressed nitrogenases in seep sediments in our earlier work was actually coming from this Seep SRB 1G. And at the time, we assumed that this was just a free-living sulfate-reducing bacteria, not necessarily connected to methane oxidation. And so we wanted to look at this also because of some of our earlier work that had shown that the Archaea are dominant nitrogen fixers within these ecosystems. So what does it mean when you have a Seep SRB partner that is also potentially contributing to nitrogen fixation in the environment? So to look at this, we use stable isotopes with 15N labeled dinitrogen gas and used a technique known as secondary ion mass spectrometry, which allows us to identify organisms by fluorescence in situ hybridization and then measure its isotopic composition after those organisms had taken up the isotopic label. So just to give you an example here, we can focus an ion beam down at the level of 50 nanometers, sputter away cell material, and then collect secondary ions to give us mass ratios of 15 to 14 in the case of looking at nitrogen. So if a cell is active in nitrogen fixation, those cells will assimilate that label into the cell, into the biomass, and we can measure that by imaging the isotopes in the cells. And so the warmer colors represent more uptake of this. So let's look at some of the data from these large ANMI2B, Seep, SRB1G aggregates here. So this is work that was done by student Kyle Metcalf in collaboration with Ranjani in the lab. And what we found was that both the ANMI2B as well as the Seep SRB1G showed evidence of active nitrogen fixation within the same Seep sediment sample. But what was interesting is that both partners did not seem to be fixing nitrogen within the same aggregate. They seem to either have aggregates where the ANMI2B were the dominant nitrogen fixers and the Seep SRB1G were less active in nitrogen fixation, or conversely, where the sulfate reducing partner was more active in nitrogen fixation and the bacteria or the archaea was not. And so we're seeing quite a bit of diversity in terms of how these organisms are partitioning in terms of nitrogen fixation among different aggregates of the same groupings in the environment. And we're still trying to best understand what's driving that. There does appear to be some relationship between active nitrogen fixation and the spatial location of cells within very large consortia, which may suggest some localized nitrogen limitation, but that can't be the only thing that's driving these spatial patterns, and why one partner is turning this on and the other is not is still quite a mystery. Okay, and so I'm just going to quickly end with a couple of slides about what we've been doing with looking at Seep viruses. And we've been doing this work in collaboration between Aditi and Alon in the lab, and looking at the morphological diversity of these viruses within the Seep ecosystem. We see there's quite a bit of diversity of these viral-like particles that were recovered from sediments, some reflective or similar to what we see in archaeal infecting viruses. And from these Seep sediments, we've created about 70 viral libraries and recovered about 140,000 biocontigs that are about 10 kb or greater, with about 3,400 completed viral mags from these ecosystems. And we specifically targeted Seep sediment samples that were enriched in different groups of these ANMI archaea, with the hopes of being able to learn a little bit more about the structure of the viral community that might be interacting with these communities here. So some that are enriched in ANMI2B, 2A, or 2C, and vice versa. And so just showing you some of the data that Alon has created most recently, looking at the full diversity of these completed viruses. What we find is that there is greater similarity of viral assemblages that are coming from the same core at different depths relative to across different Seep sediment samples that were collected. And so samples that come from one Seep location seems to be driven by the depth of the sample rather than comparing across cores that were collected at different Seeps within this ecosystem. And so from this, we're really trying to better understand what's driving that structure of the viral community relative to what we see in terms of the microbial assemblage and how these organisms might be interacting with one another. So just to conclude, we see a lot in terms of the syntrophic interactions between these ANMI archaea and their sulfate reducing bacterial partners, specifically that there is strong specificity in terms of the syntrophic partners that emerge, that exist across different Seep environments. We also are starting to better understand not only the energetics of this syntrophic association, but also maybe the unique nutritional requirements and differences in metabolic potential among different types of ANMI SRB consortia. So nitrogen fixation dynamics appear to be quite diverse among these different organisms, as well as the potential to store different types of nutrients may provide information about niche differentiation within these organisms in the environment. Looking at methane seep viruses is a relatively new component, and we see there's tremendous amount of complexity, both in terms of the morphological diversity that we're finding, as well as the genetic or genomic diversity that's recovered from these seep environments. The vast majority of them are totally uncharacterized. There's some interesting auxiliary metabolic genes that are starting to emerge, but the patterns of diversity that we see are quite distinct, going down core relative to what we see across different ecosystems that seem to have a different type of pattern than we see with the bacterial and archaeal communities in these environments. So with that, I will thank many members of the lab who have contributed to this work, as well as collaborators at UCSD for imaging, and of course, a number of different funding sources as well. Thank you.
Thank you, Victoria. That was an awesome talk. We have a question in the Q&A, so we'll handle some questions for Victoria using the Q&A forum, and then after this, we'll have some live breakouts. So the first question is, you said that you had 3,400 complete mags in quotes. So why did you use quotes? Are they not circular?
So they're predicted to be circular based on the repeats. I just, as much as we can buy into what is considered a complete viral mag, I think that's, so I think it's reasonable to say that they're complete. I know a little bit less about the genomic element of viral assembly. And so for me, quotes seems like a safer bet without being able to do sort of long range sequencing on that. Yeah.
Thank you. The next question is from Christopher, who says, thanks for the great talk. Would you expect a viral infection of one member of the consortia to collapse the entire consortia? Are the consortia balanced such that small disturbances can collapse them?
Yeah, that's a fantastic question. Honestly, we don't know a lot about the dynamics of infection of these specific ANMI and SRB associations. We do have some sediment-free enrichments now of these consortia, and we do see viruses in those enrichments, but we have not been able to show specific dynamics of infection in a way that I would feel comfortable saying this. In looking at other studies, there is some protection afforded by aggregation. So it does beg the question whether or not the cells that are on the periphery are the ones that potentially are susceptible to this. We don't see any evidence of prophage or proviruses within the genome. So it's not like they're going to be lysed from within with a stress inducer. So I think they're all wonderful questions and still need to be dug into more.
Great. We don't have any other questions in the Q&A right now, so I'll use my power to ask you a question I'm interested in, which is, have you done any work on the phylogeny of the nitrogenases from those sulfate-reducing bacteria, and does it seem like there's been a lot of horizontal gene transfer of a single type of nitrogenase among the organisms living in these sites, or is there a lot of diversity?
Yeah, that's a good question. I don't see strong evidence of horizontal gene transfer on the sulfate reducing side, and actually on the archaeal side. The nitrogenases seem to form a unique clade from the perspective of the ANMI. The sulfate reducing ones, I think, fall into the type 3 cluster, with other anaerobes. And as far as I can tell, I'm not seeing a lot of novel structure that suggests that it's being horizontally transferred from those groups. Yeah.
Great, thanks. We have some more questions that have come in. I guess I prodded people. One is from Anthony. Wonderful talk. Any insights into what drives the switching between the ANMI or SRB partner as the dominant end fixer within the aggregates? Does it correlate with sulfate or methane availability?
Yeah, we haven't tested sulfate and methane as a driver for nitrogen fixation. We have done experiments providing an external source of ammonium to look to see how that impacts. And unlike cyanobacteria that are really acutely tuned to external sources of reduced nitrogen or bioavailable nitrogen, these anaerobes tend to be a little bit more flexible. So they'll actually fix in the presence of some ammonium in the environment. We don't quite understand it. It's a little bit perplexing. I mentioned that in very large aggregates, there is some difference in the degree of nitrogen fixation from the level of N15 enrichment in cells that are towards the center of the aggregate relative to those that are on the periphery that may correspond with local nitrogen limitation by those organisms. But the thing that's unusual is in those aggregates, it's just the ANMI that are fixing nitrogen and the sulfate reducing partner is not doing anything. So we know it has the potential to fix nitrogen. I don't quite yet understand why we're not seeing both partners in the middle fixing, if that was just the answer that there was less nitrogen available. So there's some interesting dynamics in terms of interactions amongst the population of ANMI that exist within an aggregate and as well as with the bacteria that I think still need to be teased apart.
Great. Well, I think that's a good lead into this very last question. And after that, I'll let people join the breakouts. There's a link to the Zoom for the breakout sessions that you'll find in the chat. So the last question is, are there some consortia that seem split half and half by organism? Or are there some that are more intermixed, or are the organisms generally equal in the consortia?
In terms of the numbers of cells, frequently it's like 1-to-1. So if you have 500 bacteria, you'll have 500 archaea. We see differences in the total biovolume, though. These ANMI2Bs that we were just talking about, they are twice the biovolume as their corresponding sulfate-reducing partner. And in other types of aggregates, the biovolume between those two partners is more equal. And so that likely has some impact on their association. In terms of the structure and mixing, in some cases, we do find aggregates where the partners are very well mixed together. So you're almost maximizing contact with your syntrophic partner. But in others, there's quite a bit of segregation between the archaea and the bacteria. And that was one of the clues that led us down the road of looking at extracellular electron transfer, because that process allows you to relax the spatial constraints on how distant the two partners can be and still conserve energy from sulfate coupled methane oxidation. Yeah, good question.
Great. Thank you. Thanks to you and to all our speakers for this great session. And I'm sure we're set up for some lively discussions in the breakouts. So thanks to everyone for sharing your time today, and especially again to our speakers.